Hi everyone. Today we're going to be looking into Chapter 8, which is how we control microbial growth in vitro. And in vitro means basically anything that's external to the body. So to start off, we'll give a little bit of an intro. We'll talk about factors that influence microbial growth, and then factors that encourage the growth of microbes in vitro, as well as factors that inhibit the growth of microbes in vitro. So let's first start off talking about factors that affect microbial growth. The first factor is the existence of nutrients. So as microbes exist in a particular environment, they have to have access to nutrients in order to grow and to develop. Now, all living organisms require nutrients to thrive and to sustain life. And nutrients are considered to be energy sources. If you think back to our previous chapters, and our discussions on biochemical pathways, organic molecules, the generation of ATP, those basically can be summarized in the form of nutrients, right? Because nutrients are what provide energy sources to living organisms. And the only way organisms can get energy is through the breaking of chemical bonds. Now, another factor that influences microbial growth is going to be that of water. And water can influence microbes in the form of moisture because microbes aren't very big in size. So water droplets in the form of moisture help to hydrate small organisms. Water is very essential for life, and it is needed in order for metabolic processes of microbial organisms as well as larger, more complex organisms to sustain life. And one thing that microorganisms have had to adapt to is environments that contain a harsh terrain, or living in areas where the terrain is very dry. And because of this, microorganisms, at least some of them, have developed certain microbial stages that will allow them to survive the drying process, which is known as desiccation. One example is something that we've talked about in here already, is that of a bacterial endospore. Remember, an endospore is a form that a bacteria undergoes in order to survive an environment where there is extreme conditions. Either the environment is depleted of nutrients or the environment is depleted of water for the organism. Some other examples of microbial stages are protozoal cysts that also are formed whenever there is an absence of moisture in the environment. So the two key things that affect microbial growth are going to be nutrients as well as water. Now these are the two, water and nutrients are the two resources that need to be consumed by microorganisms. But other factors that influence microbial growth include things like temperature and pH change. And we're going to talk about both of these pretty extensively. Temperature is a pretty big component that influences microbial growth, and it is because every organism has an optimal growth temperature. So the temperature at which organisms grow best is dependent on the species that is living. So temperature ranges over which an organism grows best are determined by the metabolic pathways and the enzymes that are used in these metabolic pathways to help the organism to break down organic molecules or to undergo the processes of cell respiration or fermentation. Enzymes can only function at certain temperatures. Some enzymes will become denatured at temperatures that are not conducive to allowing the enzyme to function. And once enzymes are denatured, they will no longer serve their purpose for the organism. There's a wide variety of microorganisms, and all of them can reside at various temperatures. There's a lot of organisms that will grow between a range of 25 degrees Celsius, which is room temperature, as well as 37 degrees Celsius, which is body temperature. when we incubate organisms in the laboratory, those are the two temperatures that we tend to use. If you look at this diagram right here, this kind of is, this diagram here is kind of showing you a growth rate of bacteria organisms. And it looks like we've got 3 growth rates that we're looking at. Okay, so on this scale here we have the time in days, and then on this scale here we have logarithmic growth. Remember, with bacterial cells are going to replicate every 30 minutes or every 20 to 30 minutes. So that means you go from having 1 bacterial cell to having 2 bacterial cells. Now on this line we've got 3 different bacteria. We have a bacteria with an optimal growth rate of 50 degrees Fahrenheit, another bacteria that's going to grow at 40 degrees Fahrenheit, then another bacteria that grows at 32 degrees Fahrenheit. So if you look at this particular scale, what you'll see is that in the span of 2 days, the organism that grows at 50 degrees Fahrenheit is going to increase in cell numbers at a quicker pace than the organism that's growing at 40 degrees Fahrenheit. For the 40 degrees Fahrenheit to increase in the number of cells that are present, it looks like it's going to take a span of about 6 days. And if you look at the organism growing at 32 degrees Fahrenheit, it takes up to about 18 days for us to see an increase in a high concentration of cells. Now, this increase in cell growth is due to the optimal temperature at which to grow these organisms. Sometimes it takes organisms up to about 2 weeks in order to grow at optimal temperatures. Sometimes it'll take them 2 days. Sometimes it'll take them 24 hours just. So it really just depends on the type of enzymes that are present in the organism and the temperature at which those organisms thrive. Now there's some different organisms that are going to grow at high temperatures and low temperatures and then medium range temperatures. And the organisms that grow at really high temperatures are going to be known as thermophiles. And when we talk about high temperatures, we're talking about super, super hot. If you've ever been to Yellowstone National Park or some of the parks out in the Northwest, you can see a lot of pretty pools that are full of algae and other microorganisms, and they provide a very interesting, real beautiful color to these pools. But those are microorganisms, and those pools are incredibly hot at high temperatures. And so the organisms residing in those pools are actually known as thermophiles. Mesophiles are microbes that grow best at moderate temperatures. A moderate temperature is equivalent to 37 degrees Celsius or even 25 degrees Celsius. Again, 25 is room temperature, 37 is body temperature. So these particular microorganisms are kind of your common everyday organisms that we'd find everywhere. Now, because we understand that temperature influences microbial growth, we can use this to our advantage, because we know certain microorganisms like to grow at high temperatures as well as body temperature or a middle range temperature, such as the mesophiles, but there is a limited number of organisms that will grow at cold temperatures. And because of this is how refrigeration practices came to be devised, because we realized that bacteria didn't grow as well as certain other microorganisms as quickly as they do at room temperature compared to freezing temperatures, which is why our refrigerators are generally between 4 degrees and 8 degrees Celsius. So this allows us to preserve food so that way it doesn't spoil being subject to growing microorganisms. Because like microorganisms, or like us, we find a lot of nutrients as well as water from these products that we need to refrigerate, and we want to keep them fresh so we can easily freeze them to keep bacteria from growing on them and absorbing nutrients from our own resources. So temperature definitely is a big influencer. Now some other groups of organisms that we have regarding being affected by being a factor, or having temperature being affecting microbial growth, is the group known as psychrophiles. Psychrophiles are organisms that like to live in areas where it's very, very cold. So these could potentially be found growing inside of your refrigerator, but out in nature we find these growing in the depths of ocean water. Now, those are called psychrophiles, and they like extreme cold temperatures. Psychrotrophs are also a group of psychrophiles, and these are going to be kind of the organisms that you'll find living at 4 degrees Celsius, which would be the temperature of your refrigerator. And then we have a psychroduric organism that likes warm temperatures, but they're also able to tolerate very cold or freezing temperatures. So temperature definitely is a big influencer in microbial growth, and because we know this about microbes, we're able to develop practices to help preserve our food materials. The other biggest influencer of microbial growth, is going to be that of the pH. As you all know, there's this food canning process that's known as pickling, where you essentially take a very acidic solution and you store and preserve your food inside of it. And this is how we get pickles, by using storing pickles inside of vinegar. So pH is a big factor that can influence microbial growth, because the pH essentially is referring to the acidity or the alkalinity of a solution. Now, many microorganisms like to live at a neutral pH, or they might like to live at a slightly alkaline, which is also known as basic, growth medium. And if we know this about microorganisms, then we can create an environment in the laboratory that is conducive to having this type of pH, which will allow us to grow and develop microorganisms. Organisms that reside at pHs between 2 to 5 are going to be considered acidophiles, because on our pH chart, the acids are located at a pH between 1 and 5. So anything that is down in this area is going to be considered to like living in an acidic environment. Anything that likes to live in an alkaline environment is going to like to exist at a pH that is higher than 8.5. So if you look where 8.5 is, between here all the way to here, where 12 is, typically that's where you're going to have a whole lot of alkalophiles reside, because they like to live at an environment that's incredibly basic. So the pH is a big influencer on whether or not microbes will like to withstand these particular environment. And again, if we know this information, then we can grow microorganisms in an environment that's conducive to their growth patterns. The next factor that can influence microbial growth is that of osmotic pressure, as well as the presence of salt in the environment. Osmotic pressure is pressure that is exerted on a cell membrane by the solutions in which the microorganism is residing in, as well as the solutions that exist on the inside of the microorganism cell. So with osmotic pressure, what you essentially have, and here's a great example of what a cell could experience, is that you have a cell, and inside of it you have different solutes, and then on the outside of it you have other solutes. Now, in the properties of osmosis, what happens is you essentially have the movement of a solvent through a semi-permeable membrane. So our solvent is going to be either the solutes in here or the solutes on the outside, and then you have the membrane through which these solutes pass through. But with osmosis, you essentially have the movement of the water, okay? So water likes to move from an area where there is a low concentration of solutes to a higher concentration of solutes. So if you look at this diagram here, you have water moving across the cell membrane. So that